Hi again, everyone. Welcome. This is the August Recon Community Meeting. And today we're joined by Maïwenn Kersaudy-Kerhoas. I'm pronouncing that correctly. I hope I am. So Maïwenn is a professor in Microfluidics engineering in the School of Engineering and Physical Sciences in the Institute of Biological Chemistry, Biophysics and Bioengineering at Heriot-Watt University in Scotland. She's the co-academic lead for the Global Research Institute in Health and Care Technologies, and she's also a co-founder of the Global Frugal Diagnostics Network. So her lab focuses on the development of novel microfluidic tools for the separation and purification of circulating nucleic acids from blood for applications in infectious disease, oncology, and non-invasive prenatal testing. In 2012, she was awarded a 5-year Royal Academy of Engineering Fellowship to develop microfluidic systems for prenatal diagnostics. And then following this, she was awarded a $1 million EPSRC Healthcare Technology Challenge Award to further on develop these systems for cell-free DNA extraction at a point of need care. And using this award and pre-commercialization from the Scottish Enterprise High Growth Spin-Up Program, she and her team created CNA Safe, so an automated platform and patented fluidic cartridge for cell-free DNA extraction. Clearly, Maïwenn has a lot of experience in microfluidics and in developing healthcare technologies. So this is why we've invited her today to tell us a bit more about her work. So without much further ado, I will pass it over to Maïwenn to tell us a bit more about open microfluidics for diagnostics.
Thank you very much, Jan-K, for the kind introduction. I hope you can hear me well, and please let me know if that's not the case during the presentation. So I am really grateful to you and to Jenny Malloy for this opportunity. I'm genuinely excited to interact with the Reclone community and really see, what we can achieve together. And that's really, why I'm here today. So in this talk, I want to discuss about open microfluidic for diagnostics. Generally speaking, I want to talk about how do you make microfluidic devices for diagnostic purpose and what approaches can help democratize these methods so they become available to the broadest population possible. So what I'm going to do is to give example of devices that we have achieved in my lab using these accessible and affordable methods. If I have time, I will also touch upon sustainability and equity. These will lead to a few words about the Global Frugal Diagnostic Network, which was created in May this year and aim to bridge the hardware world, which is, the world I will describe to you today, and the wetware world, which I think, you guys know well. So my talk was planned for a little bit longer than is usually agreed for the Reclone meeting, but so I might pass over some of the slides. Anyhow, so I'm a microfluidic engineer, so I assume you guys know what microfluidic is, but in case you don't, microfluidics is the science and technology related to the manipulation of fluid at the microscales. And it's really the underpinning technology behind many diagnostic applications, from high throughput PCR chips like fluidine, to point-of-care diagnostic chip like the CFID cartridge for TB or HIV diagnostic, and even the humble lateral flow devices that so many of us use during COVID, because you need capillary flow, to drive the sample and the reagent through the nitrocellular strips in these devices. So the field of microfluidics has emerged in the early 2000s and has grown into many niche applications, which include things like paper microfluidics or organ nanochips or single cell applications. And the niche I've chosen to focus on is sample prep, is the pre-analytical part of diagnostic. How do you transform raw samples, such as biological fluids like blood or saliva, urine, etc., into analytes that you can actually load into a PCR reaction or into a biosensor or more complex analytical workflows like sequencing workflows. Why I've chosen to focus on that, it's because when I started my PhD back at the end of 2005, there were a lot of literature on biosensor in academic research. Loads and loads of really cool sensing modalities, optical or otherwise. The problem was many of these applications were demonstrated with ideal samples like spiked naked DNA in water, or these kind of things. And they were very far from achieving, what you really need to do, which is take a human sample and try to detect something into this sample. So I decided to focus on, on this area where, how do you take real samples and how do you transform them into analytes? And I focused quite a bit on circulating nucleic acids. So this is a new type of biomarker, which was discovered just by the Second World War, but really came to light with the work of Dennis Lowe, who found fetal DNA floating in maternal circulation back in 1997 in Cambridge, and these led to the field of non-invasive prenatal testing and the diagnosis of chromosomal abnormalities via maternal blood samples. And there are many, many more applications of circulating nucleic acids. So I've focused on how do you take samples, how do you treat samples so that they can go into an analytical workflow to analyze these fantastic new biomarkers. So you can see some examples of this work in the photos there. At the bottom, there's a blood plasma aspiration chip, which replaces a centrifugation approach to extract plasma from blood. There's a microRNA extraction chip. So microRNA is a specific type of circulating nucleic acids. There's a cell-free DNA isolation cartridge and a circulating tumor DNA enrichment cartridge. And more lately, I have focused on a little bit different things. And you see a photo there on the top right of a finger-actuated chip for blood preparation, and in particular, a filtered blood lysate. It's a very modular platform. So I cannot explain all of these different devices in detail. So I'm going to focus on the microRNA extraction chip and also the finger actuated chip. And the reason for this is because I also want to tell you about prototyping. So in my lab, we're not just focused on making devices for specific application. We're also really interested in manufacturing. I forgot to put my timer on, sorry. And in manufacturing and prototyping, that is, how to create these devices, how to make sure that you create them in a way that is reproducible, that can produce several devices that are all the same, and can produce reliable devices that can be made by other people. But also, how do you create devices that, if they work well and you want to transform them into a product, how do you do that in a smooth way, which doesn't involve redesigning completely your chip? So we're really interested in these engineering parts as well. And if you look at the examples that I've put there, and if you're familiar a little bit with microfluidics, you might think that these devices don't maybe look to what you're used to in microfluidics. And that might be because the microfluidic fabrication origins are really to be found in the fabrication technique applied to make microchips. So typically the chips, these electronic chips that you find in your mobile phones and in your computers. And that technique is photolithography. So what photolithography allows, it allows the transfer of a pattern from a photomask onto a photosensitive resin on a substrate, usually silicon or glass. But back in the years 2000s, because what scientists found that they could use microfeatures, such as the ones produced for electronic microchips, to handle cells, DNA, etc., because these micro and nano features were closer to the scale of these biological particles. But silicon wasn't a particularly convenient material. So the technique of soft lithography was developed in the years 2000 onwards. And this technique consisted of casting silicon rubber, namely polydimethyl siloxane, or PDMS. I'm sure you've heard about this term before, onto a master, enabling easy replication of micro features almost endlessly. So this technique of soft lithography became incredibly popular. I think the original paper has thousands of citations. You can replicate features in over a day, and it's reliable, but you do need a clean room, which is a very capital investment intensive. And if you don't have a clean room, you could still do soft lithography, but you need access to a master mold, to someone who can produce the master mold for you. So there's a little bit of a dependency here. And so this has been a fantastic technique for the field of microfluidics. People started developing all sorts of applications, all sorts of features, and really smart networks, etc. But it wasn't really an accessible technology because, of the need for clean room, of the need or a need for a master, for somebody to make a master mold for you. So fortunately, in the space of 10 years, from about 2005 onwards, several alternative methods came about. And the three major and most used methods that are used today are CNC milling, laser cutting, and 3D printing. And because they are using much lower capital investment equipment, these techniques are a lot more accessible to a broader group of people. And I believe this makes microfluidics much more open than it used to be. So we have CNC milling. So CNC milling is simply the old-fashioned, turning and milling that you would do in metal, but adapted to plastics and adapted to microfeatures. So the end mill can be down to 50 microns, and you can etch in a slab of plastic microchannels down to 50 microns or so. And you can make like these channels or other features that you can find in microfluidics. Now, you still need a little bit of technical expertise to use CNC. You might also have a little bit of post-processing, deburring. So burrs are these sort of features, small bits of plastic that attach after milling your part. And there are machining marks, which you can see in this figure on the bottom left, which can make the piece opaque, but it's still a very accessible and fairly low capital investment. We also have laser cutting, and my lab has done a lot of work in laser cutting. So in this technique, you work on one layer at a time, and it allows great flexibility because you can do other things like printing electrodes, adding filters, and things like that. You don't have as much freedom as something like 3D printing, but a lot less hassle in post-processing. That means once you've cut your part, you're nearly there, you're nearly ready to use your device. And you can embed a lot of channels. There's lots of integration possibility, and you can do high density circuitry. And that's what is shown in the image there in the middle, which is a map of the London tube map that we made just for fun. But we bonded 19 layers in one go for making these devices. And laser cutting is, I would say, a little bit more accessible than CNC milling. And it's a little bit easier to use, and laser cutter now are quite cheap to acquire, so it's very low capital investment. And finally, 3D printing. There's a lot of design flexibility there. These images come from a paper from Vittorio Saggiamomo, who presented the so-called escargot technique to make embedded channels. So this become really interesting in terms of 3D feature. However, there's quite a bit of post-processing that means things you need to do to your part before you can use, such as cleaning these embedded channels is a little bit difficult. And resolution is still an issue, though you can see you can go pretty low in terms of channel features. Porosity, if you're using a technique like fuse deposition modeling, which is the standard 3D printing, this can be an issue because the parts are quite porous. So if you start pumping a fluid in it or reagents, it seeps into the part. But if you're using something like stereolithography, like the escargot technique, then it's fine. And the costs massively depend on techniques. You can spend tens of thousands of pounds on really high-end 3D printers, but you can also spend just a couple of hundreds dollar, on getting fairly good 3D printers. In fact, the best, I think, is to use the best of all worlds. And if you are able to access these technologies or bring them into your lab, to use them where they are best at. So as I say, we've done a lot of work in laser cutting. So I'm going to say a few more words on these. So we didn't invent the use of CO2 laser cutting to make microfluidic devices. There were a lot of people who had proposed this technique. But what the novelty we brought about is really in the bonding. We showed that you can use ethanol, which is a very accessible solvent, to lower the glass transition temperature of PMMA, which is acrylic perspex, very common plastics. And so you lower the glass temperature, and then you apply a little bit of pressure and a little bit of heat, about 70 degrees or so, and you're able to bond up to 19 layers, maybe more, in just 3 minutes. And what this means, it means that when you have a design on your computer, go to the laser cutter, cut the chip, and then clean the layers, bond your layers in just 15 minutes. Again, as I say, if you have your design in just 15 minutes in your hand, you can have a chip that you can go and try. And then in the same afternoon, you can revise the design and make a new device that hopefully will work better. So for my lab, it has been incredibly empowering. I believe that this is a very accessible technology, is very cost effective. I would say it's reliable down to 200 microns in terms of channel width. If you need to go lower, then maybe you have to use other either other type of laser cutters such as femto laser, but then it becomes very capital investment intensive. So we have put a lot of documentation to enable others to replicate these methods. And on our YouTube channel at Microfluidic MKK, you can find a full video that takes you through the different steps. And there's a paper that details also all of the steps. And then we have put a lot of examples of chips we've achieved with this technique. And so that's what I'm going to do now. I'm going to take you through a few examples of microfluidic chip that we have produced using this laser cutting technique with the solvent assisted bonding methods. So the first example is a set of sort of standard microfluidic functions. So the first one that you see there on top left is a mixer chip. So in microfluidics, you have so-called laminar flow, which means that mixing happens only by diffusion, which can be quite slow. So people try to incorporate features such as split and recombine features to bring the fluids together and increase agitation and enable faster mixing. And so that's what we've done in this chip there, bringing a yellow dye and a blue dye and showing that it becomes green, and when you increase the flow rate, the agitation is higher and you have more diffusion happening, and then you can mix quicker. Then we also have developed a H-filter chip. This is another really bog standard microfluidic concept. And the idea is to bring together two streams of fluids, and again to use the very controlled diffusion in order to separate one compound from a particular stream that you introduce in the chip. So what we've done here is that we have flown a stream of water with a stream of fluorescein and blue dye in water. And what happens is that fluorescein is a smaller compound and it diffuses much quicker into the clean stream of water. And so at the end, you can get all your pure fluorescein in water on one end and the rest of the unseparated mixture on the other. And finally, we've developed also a droplet chip. So droplet encapsulation has been used in microfluidics a lot. It's used to encapsulate anything from DNA all the way to nanomolecules, all the way to things like nematodes, little worms, so that you can apply, reagents or test the efficacy of different compounds on them individually in a sort of very combinatorial manner. And so what we've done here was, just for fun, we created this complex Celtic device just to show off, our technique and how intricate the features can be. And we've created this T-junction that you can see here that brings together a channel which carries water, which is the sample here with red dye, and a stream which carries oil, which is the carrier here for creating droplets. And you can see that at different flow rate ratios, you get different type of size of droplets, etc. This is of great interest for people who handle droplet in microfluidics. We haven't characterized extensively, but these just show you can get different size of droplets. And typically this device can be done in just a few minutes, which is quite cool, I think. The second example is a chip for cell-free nucleic acid extraction. So here the use case, I want to tell you a little bit about the use case, which is around drug-induced liver injury. So here we had a real sort of biomedical needs, and this need is the need to detect patients that have gone through paracetamol drug overdose. So paracetamol drug overdose is a very common reason for patients to come in A&E. And at the moment, we only have the mean of ALT and AST level to detect if there's a drug-induced liver injury. But this is not really precise, and sometimes it happens that hospital will release patient home because the ALT and AST level are quite low, and unfortunately, this patient die at home because the marker has not detected the liver injury. So one of my colleagues, a toxicologist at the University of Edinburgh Medical School, found a new biomarker, which is a microRNA 122, which we hope is much better at detect drug-induced liver injury. Typically, it shoots 100 times above its normal level when there is drug-induced liver injury. And so what my colleague wanted is a system where you would take a blood sample from the patient, do plasma aspiration, microRNA extraction, and then an electrochemical-based detection in order to quantify the level of microRNA 122. And so my lab helped to design a chip that would extract microRNA from the sample for then to pass on to a colleague who was doing the electrochemical detection. And so here you see images of this chip that we have created. In the various chamber, we can load reagents, typically, the wash, bind, elute reagents you have for DNA extraction. And there's a series of membrane, the typical silica membrane that you use for nucleic acid extraction, but it's all encapsulated in a microfluidic chip, which means that you can load all your reagents, place this cartridge in an automated equipment, and then press the start button and let your system running and get the extract of microRNA at the end. So you can see the scale of the chip on the photograph, and you can see the different layers that we've used with laser cut in order to put the system together and achieve microRNA extraction. So here are some of the results that we've got. So we've looked at a panel of different microRNAs, and we have shown that there was no differences between the eluates from our microfluidic cartridge and the eluates from a bench, like a normal bench extraction, so somebody pipetting in and out of silica columns, apart from a few microRNA that shows a difference, but not microRNA 1,2, which was of interest here. So what we've done is also look at healthy plasma and also marking healthy and disease. And we have found that we could see a difference between the marked healthy and the marked disease following extraction from the cartridge. And we also compared to the bench and didn't see major differences between the bench and the cartridge, which is really good and shows that our cartridge is robust enough to achieve the same as what a human would do on the bench. We also looked at following the CT values from the cartridge and from the benchmark, and we had a good tracking behavior there. And when we put this eluate on the electrochemical sensor, we also see that we could track values between the benchmark and the cartridge. We did acquire some clinical samples, and we showed 75% sensitivity and specificity on a small range of clinical samples using this chip. Okay, trying to move to the next one, but I seem stuck. Apologies for that. Okay. So my third example is a modular so-called frugal device for blood sample preparation. So here the use case was around a clinical, the clinical need to measure immunosuppressant drug prior to surgery. So the clinical transplant surgeon contacted myself and my colleagues at Heriot-Watt, and what they wanted is the ability to measure tacrolimus, which is an immunosuppressant drug, in blood samples prior to surgery. His problem was that he injects this immunosuppressant drug into his patients, and then he take a blood sample because this drug has a very narrow therapeutic range. And so the range has to be, very precise in patients, and it's hard to calculate beforehand. So what happens is that he sends these samples to a detection with mass spec. It takes 24 hours. The patients come back, and then he has to adjust. And so what he wants is a chip where he would inject the samples into the patient, then take a blood sample, then analyze directly, and then do this adjustment directly, and then go to the theater to do the surgery, instead of waiting and wasting precious clinical time. So what he wanted to do is take, from a blood sample, lyse the blood samples to release the tacrolimus bounded to the red blood cells, and then filter that so that it can go to a sensing mechanism, whether it is an integrated mass spec or other type of sensing mechanism. Okay, sorry, I'm again stuck every slide. Apologies for that. Okay, so we came out with an idea to do this in an integrated microfluidic function. And I'm going to take you through this chip because we really packed a lot of little innovation, little functional things, very sort of frugal and and that we're happy to share with people. And we think that it's sort of a modular approach which could be transformed to do, other type of functions for diagnostic devices. So what you see here on the left is a CAD drawing of the chip, and it has a number of functions. First of all, it has a storage function. So we use blisters that contain the lysis agents so that it can be on chip, and the person using it doesn't need to load anything in. They just use their fingers to crush the blister and release the lysing agent, which is going to lyse the red blood cells to release this tacrolimus. We've also come up with a metering valve, which enables us to meter exactly 15 microliters of blood. The third function is an interfacing function. So we have shown that we can use these mini-vet point-of-care devices to collect blood and then push the blood inside this device and then use the metering function. There's also an acoustofluidic chamber for mixing the blood. So it's a very simple device that uses a piezo element, which is a very common microelectronic component. And then above the piezo, what we have is a slab of silicon, which has little holes. When you put liquid in this reagent, in this reaction chamber, the little holes keep air. And when you actuate the piezo component, what happens is that the bubbles extract and contract, and this creates mixing and enables you to do things a little bit faster. We also have a simple filter to remove any debris and an optical interrogation zone where the tacrolimus, this drug, will be detected. And finally, a large volume displacement pump, which is a no power pump, which enables you in just one go to empty this reaction chamber and push your mixture through the filter and into the optical interrogation zone. So you can see on the right image which shows the different layers that we've used for this device and different little components. And I'm going to take you through a number of these functions. So I'm trying to, actuate the video. So here we have a little video of the blood that we've injected into this chip with the lysing agents. And when we turn on this piezo actuator, you can see that we can mix the blood and the lysing agent really, really quickly compared to what would happen in a tube, for example, and without the need of some depipetting in and on. And we have tested this into very high hematocrits, typically 65% hematocrits at most. So here are some more videos. So here is the illustration of the reagent storage in this blister. So what you're gonna see is somebody loading just manually for the demonstration of the blister, just a liquid, and then you see the finger crushing the blister and putting these reagents together. And then on the right, you see the manual metering valve, which enables to meter exactly 15 microliters of blood. And this is really simply made. It has a 3D printed shaft and a molded silicone housing, and that fits into the main cartridge. And then you can turn manually in and out. You can retain liquids, and you can decide when to release a liquid. Underneath the seat of the blisters, you have simply an engraved needles and a little hole. And what happens is that when you push on the blisters, these little needles, they enter in the blister and they crack it and they release the reagent through this little hole. We've also invented this no power pump, as I said. So this is a really little funny story. So just by complete serendipity, the postdoc who was working on this device was using electronic cigarettes, and there was a little cap on the cigarette, which was in silicon. And he noticed that this would enable enough pressure, if pushed against a little microchannel, to empty a reaction chamber. And so he simply took this device, pierced a hole in it, and then created a ridge on the chip to fit the little silicone cap. And then by just pressing it, he showed that he was able to empty this reagent chamber. So it was really simple, very efficient, and extremely cost-effective way to have a no power pump into a microfluidic device. And so finally, here is the image of the device assembled. And here we have a little video of the device being used by the postdoc to prepare a blood sample into a filtered blood lysate. I won't show the entire video, but you can watch the videos on the YouTube channel if you're interested. So the device was so simple to operate that one of my team members suggested I brought it home to my daughter, my 10-year-old daughter, and tried to make her use the device. So what I did is I created a series of 8 simple instructions to use the device, and I just didn't say much. I gave her the device and these images that you can see here, and 8 line of instructions. Then we made it a little up to 11, and she operated the device herself. I'm just going to show you a little bit of this video. I hope the sound is okay for you. But here she's loading the sample, not blood because it was at home, and she's piercing the blisters and releasing the reagent. And then at the end, she's going to use this pump, and you can see how reliable. So at first she didn't manage, she was pushing really hard and didn't manage, but then eventually, she managed to get the sample where it's meant to be. And so really this show that these functions are super reliable if you can take them out of your lab and get your 10-year-old daughter to use them. So following on from these, we actually shipped a few of these devices around the world, for people to use the different functions as they wanted. So a goal in my lab is really to democratize microfluidics, and I didn't start with this goal. I just found out by serendipity that the techniques that we were developing can actually be really useful for people. So in 2019, just before the pandemic, I was invited to give a talk in Peru and deliver a hands-on workshop on microfluidics. And in one afternoon, with the very nice smiley people that you see here in this photo, we created a droplet encapsulation chip. And so people were really excited. And one of the attendees of the workshop, Pia Padilla Humanintinko, published this photo on Twitter just like a week or two later. And he basically had replicated, the little hands-on workshop that we had done together in his lab. And then I realized, how useful all of these techniques can be to people, and how empowering, and how they can really democratize, under the words of José Gómez Marquez, radically democratize the tools of medical creation.
Okay, I think we just lost Maïwenn.
Oh, I think
sorry, you're
just
muted again.
back. Yes.
Yeah, I'm back. Apologies for that.
No problem. Take your time.
So yeah, we have a little bit. Can you hear me?
We can hear you, but you might need to share your screen again. I think we lost that.
Yes.
No worries. But take your time, Maïwenn. Don't worry.
Apologies, really sincere apologies for that.
Not a problem. Right, you're back. Perfect. Thank you.
So what did you hear me going on for sustainability?
No, I think we just lost you there. You were just finishing talking about the workshop with Pierre Padillo.
Yes, yeah. Yes. So basically, I think these methods can democratize access to microfluidics, and that means democratize access to diagnostic innovation, essentially. Now, I had a whole part on sustainability, but I'm a little bit short of time. So I think, I just want to highlight that there's a lot of plastic in my devices. There are a lot of plastics in the commercialized diagnostic devices, whether there's the CFI cartridges or, these LFA tests, which are produced in billions. And there are many drivers for the reason for why we're using more and more of these devices, and there are some good reasons and there's also some bad reasons. The environmental impact is very much locked at the design phase, which is why I think I, as a designer, as a microfluidic engineer, I have some responsibility to look at the full life cycle of the devices. So not just, the function, but what are the materials that I'm using and what people will do after they've used the devices. So we have, together with a lot of colleagues, including colleagues at the African Society of Laboratory Medicine and MSF, we've published a perspective in the journal Lab on a Chip, looking at the sustainability of point-of-care diagnostics, and making a series of recommendations and highlighting the work that people have done, using recycled plastics or using bioplastic or using natural fibers. And there's a lot of potential for sustainable diagnostic in paper microfluidics, for example. Unfortunately, these are still quite low technology readiness level and haven't been commercialized. We have also in our lab, we've spent a lot of time doing things in bioplastics, which is a good material if you have to incinerate at the end of your life, and if you have to have high hydrophobicity, etc. And we've engaged medical diagnostic manufacturers, microfluidic manufacturers, raised their awareness and shared our results. And hopefully these commercial suppliers will start selling, other type of materials than virgin plastics. We've put together a whole roadmap on things that can be done in diagnostic to make them more sustainable. And in this journey, we have also uncovered another problem, which is that all of the IP around these devices is in the global north. And there's very little financial equity. Health equity has been constructed around the access to diagnostic, but we'd like to put another concept here, which is the financial equity and the possibility for population to actually make revenues, out of diagnostic devices, and therefore they have to innovate, and develop strong R&D ecosystem to do that. And we start, putting some ideas on how this would happen. To a certain extent, there has been recent progress. Organization like PASS, advocate for diagnostic companies to be local to their target markets. There's a drive towards local manufacturing. We've seen these with Diagnostics being the African Senegalese manufacturers of lateral flow assays, and Reclone, which bring independence to reagent productions and a lot of teaching programs. And the question is, what can be done beyond this to accelerate this ecosystem of diagnostic innovation? And so together with many colleagues, including Heriot-Watt and Elena Roska, which you will all be familiar with, we've created a global frugal diagnostic network to increase access to point-of-care diagnostic developments, with a little bit of funding from the Royal Academy of Engineering and support from a number of universities. So what the Frugal Diagnostic Network want to achieve is really to see what can be done to promote R&D and enterprise creation in diagnostic, especially in low and middle income countries, and what can we do to create this ecosystem. In particular, we'd like to leverage the Fab Lab networks, which already has these things like 3D printing, laser cutting, CNC milling, etc. What can we bring to these Fab Lab networks so they can start innovating in the field of diagnostic? And can we share good practices and standard operating procedure in these area to accelerate innovation? So we've just launched in May this year. We had our first meet and greet, and at the end of this month, we'll have our second meet and greet with Professor Kwabena Duedu and Professor Till Bachmann as our main speaker. So I would really encourage you to join the network. And what I want to do, and I'm happy to start discussing. I'm not sure how much time we have now. But what can we do to bring the wetware network with the hardware network to create new ideas, integrated prototypes where we bring in, the new enzymes, the new probes, the new markers together with, hardware that can make this technique more accessible. So I will stop there. Thank you so much for your time. And these are some of the people that enable the work I've presented today. And I'd like to thank Harry in particular, who's been really helpful in connecting me with the community of Reclone and many other communities, and also my colleague Alice Street, who is a social scientist focusing on diagnostic, and with whom I've developed a lot of the ideas I've presented today. Thank you very much.
Thank you. Thank you so much for an amazing presentation, really inspiring work. Thank you so much for sharing with us. Please make sure to share with us also about the meet and greet you're having later this month. It would be great to have it on Reclone Forum. We'll for sure try our best to be there, and I'm sure a lot of people here in the audience would like to join as well. Oh, yes, and Yankei just shared your YouTube channel for everyone that is interested. Oh, thank you. And then we just got the link to join the Frugal Network, Frugal Diagnostics Network meet and greet. And I see we have a raised hand from Kenny Timi. I hope I'm saying your name right. Please feel free to ask your questions.
Oh, great. You got the name right. It's not really a question, but I just wanted to really commend the speaker, Maïwenn. I never knew that she'll be the one giving the presentation today, because I also am part of the Global Frugal Diagnostic Network. So I want to join voices with her to invite everyone here to say they should show up for the August meet and greet. She did a fantastic presentation, I must say.
Thank you very much, Kenny Timi.
You're welcome.
Great, thank you. Yes, we also had a comment exactly about your amazing work. In one of the slides that you show, that it looks like a miniature factory. Yes, it's really like something. And it's for me is also new. And well, do we have any questions from the audience? Please feel free to raise your hands or write them on the chat.
So indeed, Microfluidics, through the miniaturization enables you to create mini factories. Here it's all illustrated in the field of diagnostics, where you can essentially sort of miniaturize a centrifuge and, a DNA extraction, the vacuum ring that you use, and all of the pipettes, and maybe even a qPCR, all done, into miniature chip. And that's what the C5 cartridge is like a mini laboratory into a small piece of plastic, essentially. And this can also be true in the pharma industry. There are microfluidic chips to make, pharmaceutical compounds. It's also used for environmental diagnostics, veterinary diagnostics. There's many applications.
Awesome, yes. And well, I invite again everyone to check the YouTube channel we shared on the chat. You will probably see a lot of these devices. That's awesome. And yeah, so we have a question on the chat from Victor. What is the biggest challenge you have encountered in creating microfluidic systems in diagnostics?
So as an engineer, handling biological fluids has been quite a journey and a discovery journey, because biology just changes all of the time, and it's so complex, and the blood sample is so complex, and is a challenge in itself. I think that in general, the challenge is to make things reproducible. And so you have really benefited from establishing standard operating procedures, SOPs, and really detailed SOPs so that you can really make, Microfluidics in a reproducible way. So that is a challenge, and you sort of need to be organized in order to do that, because otherwise it's easy to go to the laser cutter and produce a chip, and the results. But if you haven't recorded every parameters you've used, the next day, you're going to make a different device and it might work differently. So I would say that it's a challenge that comes in my mind.
Oh, definitely. Yes, I think this might be a common challenge in many different areas to make sure it's reproducible. Other scientists, other people from over the world can continue your work. And we saw a great example from the workshops you are already helping organize in different countries. So that's amazing. And we have a raised hand from Edgar. Please feel free to open your microphone and ask your question.
All right, thank you so much. I'm Edgar Tamba. I'm a research assistant here at Makerere University in Uganda. So my question is about actually the infrastructure. Thank you for that presentation, but in a setting like Uganda, I'm not sure how we can be able to, pull off such devices. So in terms of infrastructure, how easy is it to, set up a lab that can start off, such things? Because to the least of my knowledge, I'm not sure if we have one yet. Biomedical engineering students actually studying topics like this.
Yes.
So, yeah.
Thank you so much for your question, Edgar. Much appreciated for you to raise it. I can put you in touch with two researchers at Makerere who have a background in microfluidics and who are producing some kind of microfluidic devices. So the method I present here, essentially what you need is a laser cutter and a hot press. A hot press, we have used, the first one we used was just a few $100, something like that. And the laser cutter is a little bit more than that, but now you can acquire a laser cutter for, I don't want to make mistake, but maybe $2,000. Maybe a little bit more. These are not going to be absolutely the best, but they would enable you to start going and to start playing around with devices, with designs, etc. And so with that, you would be able to recreate the devices I have shown in there. And I'm in touch with Makerere University to also help them progress in their curriculum. So they've put together a curriculum for microfluidics and MEMS, so micro electromechanical systems, and they will start, I don't know exactly what kind of equipment they have, but I know that they want to grow their equipment and their capabilities. And so it's really an exciting time to ask this question. And if you drop me an email, we can continue the discussion and see what we can do together. And this is exactly what the microfluidic network is about. It's about helping people to see what equipment they need in order to prepare these devices.
Great. Thank you. Yes. And we already see the network in action. Edgar, do you have any other questions? Okay, perfect.
Sorry, thank you.
No problem. Then we have a question on the chat before going back to the open microphone from Joseph. Can microfluidics chips in medical diagnostics cancel the need of large blood samples and maintaining the accuracy?
That's a really good question. I don't know if people have heard about oh no, the name escaped me now, but this woman in the US who started a company, I'm sure somebody can put it in the chat, it's just my mind went blank, and she promised that she would do 100 tests on a droplet of blood and that there wasn't the need for normal venisections, etc. Theranos, thank you. And she failed miserably because there's sort of a physical limit to what you can do in a droplet of blood rather than milliliters. In my practice, we've always gone for large volume of blood, typically in the order of a few milliliters. So something you would, take from the arm rather than a finger prick. And that's because we're interested in cell-free nucleic acids, which are in low abundance in the blood. But there are a number of other medical diagnostic that you can do with just a finger prick. I think the field of microfluidics had the big ambition that we would be able to massively reduce the volume of blood. I don't think we've quite realized this yet. And this is because blood samples are complex, you need to clean them, then you lose some biomarkers, and then you don't have enough to do your detection. So there's been a little bit of a backwalk on these, backtracking on these, and going back to fairly larger. But in the principle, yes, you can reduce blood samples, and there are a number of examples out there.
Great. Thank you so much for sharing. We have a question now from a raised hand from Ordi. Would you like to open your microphone?
Hello. Hi. Can you hear me?
Yes.
Great. Hi, Maïwenn. Thank you very much for the presentation. It was very exciting seeing everything. These are so my name is Ordi, and I'm a research fellow at Ateneo de Manila University in the Philippines. And our group has been doing microfluidic device prototyping as well for the past few years. Actually, part of it is largely based on your work as well. So the 2016 paper about acrylic. So my question is a little bit around that, but I also got a bit distracted by Edgar's question because similarly, we've been able to carve a little niche here in, I think, the Philippines for laser cut and CNC milled microfluidic devices. And we're trying to grow the lab a little bit, trying to teach people how to do our fabrication methods, and also trying to get them closer to, I guess, best practices around the world. My background is also in engineering, and I'm also kind of diving into biology now because of this experience. So I was hoping to ask a similar thing in that I actually have two questions. So the first one is a bit more in the weeds. A lot of your work, the devices that you presented today are in acrylic or PMMA. Have you found the need to pivot to a more biocompatible material, or has it served you well so far?
Thank you so much for your testimonial, Ordi. I'm much appreciated to hear that. It's fantastic. It's what all researchers want to hear. So yes, so we haven't felt other than sustainability, we haven't felt a strong need to move from PMMA. In terms of handling blood, doing all of this sample prep, I think it works really well. And we haven't found that we are losing, a lot of cell-free DNA by doing that, for example, or cell-free nucleic acids. It's such a agile material. It's so easy to get, and it's very easy to use, to clean, etc. So it does. It's transparent. With a little bit of modification, you can do cell culture. So yeah, no, it's quite an agile material. Now, in terms of sustainability, we have tried to move to PLA, but the problem is that we can't buy slabs of polylactic acid, which is a naturally derived biopolymer. So we have to make our own, and then it becomes very hard to make them super flat and then laser cut and get good results for bonding, etc. And the bonding methods have to be changed as well. So we're still not there in moving to a bioplastic. However, I'm more and more interested in paper microfluidics. People are doing fantastic work around the world in paper microfluidics. I think what we need to see now is commercial devices based on paper. But yeah, to start in paper, it's almost like it feels like starting from scratch again. So we're sort of pushing back the dates we're going to do that.
Thank you. My second question is about if you've ever explored larger scale production of the acrylic devices, maybe injection molding.
Yes, I didn't touch on these at all because I didn't have time to go into everything, but we did. So this chip that you see here is a molded chip. It's molded in 3 parts. It has a part that keeps the reagents, a fluidic part, and then just a part in intermediary part that holds these 2 components together. And so what we've done is, once we were happy with this design, with the principle, and how we were adding the reagents for the nucleic acid extraction, we moved to injection molding. And I'm doing this more and more. I've also done some consultancy for others to mold their parts. We don't mold in PMMA, we mold in COC, and that's really driven by just the microfluidic industry being keen on COC. Because you have really good transparency, and all their QC, their quality control methods in this company are based on optical detection. So they favor COC, which is a little bit more expensive material, but then you can also mold, you know, in polystyrene, and you can mold in acrylic if you need to.
Thank you very much.
Thank you, and nice to meet you, and let's keep in touch, and please come to the Global Frugal Diagnostic Network.
Perfect. Yes, I think we have time for one more question that is already on the chat. So from Hanif, thank you for the nice talk and really nice to meet you. Is there any work that your team has done for biofuel cells or enzymatic biofuels for implantable devices?
Thank you, Hanif, for your question. Now, I'm not sure I completely understand what is an enzymatic biofuel for implantable devices, but I'm going to say, one, we don't do implantable devices in my lab, but actually PMMA acrylic is biocompatible, and yeah, anyway, there's a cool story around that, but maybe we don't have time. But I mean, people have, as I say, you know, people have looked at reaction monitoring and chemical reaction, miniaturized chemical reactions. So I think, you know, I'm not exactly sure what you mean there, but if you know, just, you know, put in a search engine, biofuel cells and PMMA, you will find loads and loads of literature on this.
Great. So thank you everyone for all your amazing questions. If you have anything else, there is already a thread on our forum where you can ask more questions. And yes, as Yankee just shared, it's really lovely to see the connections between the different research groups that we saw today. And as I said, please keep engaged with us on the forum. Yankee also shared the link for our discussion there. And of course, the invitation to join the Global Frugal Diagnostics Network meeting later this month. And of course, we also have a special invitation from our side. I'm going to just share with you here. So we have our upcoming Recon Forum is actually the 6th forum we haven't done a forum. I'm sorry, symposium forum kept in my mind, but our symposium, we haven't done a symposium in a while, and we're really excited to be back with all of you. So we already have a save the date on our website. So our symposium is happening on October 11th. It's going to be a bit of a longer session than our monthly meetings, and it's going to be focused on building scientific communities through open science research, education, communication, and collaboration. And until we get to the symposium, which we should share about registrations pretty soon, you can also check some of our previous symposium recordings here. And of course, as someone asked earlier about previous meetings, all the recordings from our meetings are also available on our website, and you're all invited to check. But I wanted to make this special invitation for our upcoming symposium. And yes, thank you everyone for joining us today. It's always lovely to have everyone here. Thank you so much.
Thank you again for the invitation. It was lovely to meet everyone. Good day to you.
Thanks,
Thank
Gary.
you.
Thank you, everyone.